So welcome back again to part 3 of the revision, physiology practical revision in preparation for the steeplechase exam, MB exam. So in this section, we'll be talking about bleeding time and clotting time. Like I would like mentioning it over and over again. Is a steeplechase practical. You're not going to be performing the actual practical. We've done the actual practical in class, hence why I am not performing the actual practical. Why I'm doing more of talking and showing of the equipment and doing more theoretical aspect, because you're going to be doing a lot of identification and or whatnot. So for the bleeding time, I want to start with the equipment and apparatus that we use for bleeding time. I've shown it before. We use the sphygmomanometer, and we use sphygmomanometer. We use lancets, sterile lancets. We use filter paper. This is what it looks like. Filter paper. We use cotton wool and methylated spirit. So, and you have to know that there are two methods of bleeding time that it's being done. We have the Ivy's method and we have the Duke's method, and I'm going to explain the both of them as we progress. For the Ivy's method, I'm going to give you the normal value for the Ivy's method. For the Ivy's method, the bleeding time lasts between 2 to 7 minutes. Please note it. Ivy's method, the bleeding time lasts between 2 to 7 minutes. While for the Duke's method, the bleeding time lasts between 1 to 4 minutes. I hope I'm making sense, and I hope you are getting it. Okay. For the Duke's method, you will use either the fingertips or the earlobe. All you do, you prick the finger, you prick it, either the finger or the earlobe, and you start your timer and start blotting after 30 seconds. After 30 seconds, you blot. By the time you blot with using your filter paper, at the end, when there is no blood stain on the filter paper, you add up the 30 seconds, which gives you your bleeding time. It's almost the same thing as the Ivy's method. In physiology lab, we use, in my lab, we did what we did. You can remember that we did the Ivy's method. For the Ivy's method, you use the sphygmomanometer. You inflate the breaker. You inflate to 40 millimeter mercury, and you make a standardized incision, and you start your timer and you blot after every 30 seconds. At the end, where there is no more blood stain on the filter paper, you stop the timer and you add up the 30 seconds. That gives you your bleeding time. I don't know if it's clear. Okay. So there are clinical significance of bleeding time. Bleeding time is prolonged in certain conditions such as thrombocytopenia, platelet dysfunction, aspirin use, vascular disorder, and von Willebrand factor. So they could ask you, state the conditions in which bleeding time are prolonged. I said platelet dysfunction, aspirin when you are using, or the subject or the person is using aspirin, when there is vascular disorder, von Willebrand disease, which is a very common one that people, you've heard that a lot. We also in thrombocytopenia. These are the conditions where bleeding time is or can be prolonged. So we move on to the clotting time. Time is basically the time it takes for blood to clot after removal from the body. It reflects intrinsic and the extrinsic common coagulation pathways. I don't know if you understand. I said clotting time is defined as the time it takes for blood to clot when it's outside the body. Yes, our blood does not clot when it's inside. It's when we withdraw it and put it in container. When it's outside, the time it takes for blood to clot is known as the clotting time. And this shows the how good or how functional the individual's intrinsic and common coagulation pathways, how they function, if they're in good state. And there are two methods of clotting time or coagulation time. We have the capillary tube method and we have the slide method. We've did the both of them in class. We've seen the practical aspect of this. This is just a revision to remind you of what it looks like, because like I said earlier on, it's a staple chain. All you need to do is identification and filling the possible gap. So for clotting time, we use for the apparatus that we use for clotting time, we use non-heparinized capillary tube because we want to check how long it takes for blood to clot. And we use the slide for the slide method. We use capillary tube for the capillary method. We use the lancet, we have methylated spirit and cotton wool. For the capillary method, you sterilize the finger. Most I will use the middle finger. You sterilize the fingertip and you fill up the capillary tube. And every 30 seconds you break, watching for the fibrin strand. You are looking out for the fibrin strand. At the point, recording, after every 30 seconds you break. After every 30 seconds you break a little portion of the capillary. This is the capillary tube. So after every 30 seconds you break a portion. After every 30 seconds you break, looking out for the fibrin strand. By the time they see the fibrin strand is visible, you stop the timer and you add up the 30 seconds. That gives you the bleeding time for the capillary method. Why for the slide method, you get a clean slide. Make sure the slide is clean and free from all form of impurities, and you pierce and drop a little blood on it, and you can use the capillary tube to stir and raise. You stir, you raise, and what the purpose of this stirring and raising it up is to be able to know when the fibrin strand has formed. So at the point where you can see the fibrin strand, you will see it. I showed you in class, you saw it, we did it, you saw it. You raise it up, you see it, you stop the timer, and you add up the 30 seconds, and that gives you the bleeding time. So either ways, we did the both of them, you saw the both of them. So either ways is the clotting time. For the clotting time when you are using the capillary tube is shorter. We have between 5 to 11 minutes. While for the slide method, we have between 8 to 10 minutes. When you write either of this one for the exam, you are good to go. Just know that. And okay, next thing I need to talk about is, sorry, I'm trying to, so that I don't miss any of my points. I jotted my points down, so that's why you see me looking at somewhere. So why is clotting time important? Clotting, there are some factors, there are some conditions, physiological conditions that can lead to prolonged clotting factor. Some of these are like, example of them is we have the hemophilia, liver disease, vitamin K deficiency, and anticoagulant use. These are the conditions where blood will not form a clot. We know the importance of, I've explained the importance of this clotting factor and bleeding factor in class, but I just want to give one or two here. Why clotting and bleeding time that are very important is because if there's any abnormality with the clotting and the bleeding time, when a person faces a cord or have a cord or probably get involved or have any situation that will warrant them or lead them to bleeding, there's going to be a problem, because they are deficient of the factors that can lead to stoppage of the bleeding time and bleeding and formation of clots. Hence, when somebody, maybe when that individual, let's assume the individual have an accident and there is a deep cut, if care not taken and the person is appropriate medical attention is not given to that individual, the person could bleed or can bleed to death. That is why bleeding time and clotting time are important. So when some of these things are being done, we do it in the physiology lab, but this is for teaching purpose, not for teaching purpose, not for diagnostic purpose. We're only showing you what it looks like. There are other tests that could be done in the medical lab to ascertain and to confirm if all these things is actually there. So the difference between bleeding time and clotting time: one, bleeding time measures the platelet function. Bleeding time measures the platelet function, while the clotting time measures clotting factor activity. You've been taught everything. I know you've been taught all these, the theory parts, so I don't need to go in deep into it. And we also talk about the bleeding time. The normal bleeding time is between 2 to 7 minutes, while for the clotting time is between 5 to 11 minutes. And bleeding time is prolonged in platelet function, platelet issue, when there is an issue with platelet, while clotting time is prolonged in factor. We have about 13 factors that cascades that leads to one till you get to blood clotting occurs. So clotting time is prolonged when these factors are deficient. Yes, when these factors are deficient. And when you're performing this experiment, you perform them in room temperature. Make sure that the environment is conducive and the atmosphere at 37 degrees Celsius, like the same temperature with our body system. So I think that is it for this. If you have any question, please, you can drop it in the comment section and I will answer you. So, okay, see you in the next practical revision class.